Hello, my name is Emma, and I chose to do my project on Noonan syndrome. So, Noonan syndrome is a congenital genetic disorder. Its common characteristics are distinct facial appearance, broad or webbed neck, low posterior hairline, chest deformity, and short stature. And a lot of the characteristics are physical, but there are some internal as well. So, some more physical are facial features that include widely set eyes, skin folds covering the inner corners of the ears, drooping upper eyelids, a small jaw, depressed nasal root, short, broad based noses, and a low set rotated ears. It also includes some skeletal abnormalities like sternum issues, spine curvature, and outward bent elbows. And then internal, there are some heart issues that come about. So there's pulmonary valvular stenosis, hypertrophic cardiomyopathy, issues with blood and lymph vessel malformations, clotting, platelet deficiencies, learning difficulties, things like that. I chose this condition because I actually saw it diagnosed in a medical office that I was shadowing at, and thought it was sounded really interesting because it was rare, and then there was a lot of information on it as well on NDX, so that was perfect. So, systems biology, what is it? Systems bio, it focuses on the study of complex biological systems as integrated networks of genes, proteins, and molecules. The goal of systems bio is to understand the behavior and functions of biological systems. So, rather than studying individual components in isolation, systems biology takes kind of a more comprehensive approach, examining how different elements within each biological system interact with and influence each other. Why is it appropriate for this disease? Noonan syndrome is a multifactorial disorder. So, because of this, systems bio allows for the study of the interactions involved, considering the integrated network of various genes, proteins, and other molecules that are involved in the RAS MAPK signaling pathway, which is the primary player in the cause of Noonan syndrome. So, systems bio uses network based approaches to analyze relationships between different components of biological systems. In Noonan syndrome, mutations in genes such as these three, or especially SOS1, contribute to dysregulation in the RAS MAPK pathway. And so, network analysis can help figure out how these mutations affect the overall signaling network. So, my analysis of the paper I chose looked at the difficulties regarding the incorporation of biological relationships between biomolecules into unified data models. So, it talks about differences between physical interactions and causal interactions. Um, Previous studies in protein interaction databases focus mainly on capturing physical interactions. Um, because representing causal interactions has shown to be more challenging because of the absence of sort of a standardized model. In the article, two approaches were covered, the first one being process descriptions and the second one being activity flow diagrams. Process descriptions offer insight into various mechanisms, but it poses challenges for practical analysis. And then activity flow diagrams simplify representations by highlighting protein activities connected by activating or inhibitory edges. So they're less detailed, but these diagrams work better for larger models. This is the data set that I chose to focus on. Here at the bottom middle, you can see the node that I chose as well. It is SOS1. So here I have the node in the pathway. So SOS1 is a node that encodes a guanine nucleotide exchange factor responsible for stimulating the conversion of RAS from the inactive GDP bound state to the active GT bound form. The pathway that I've focused on for this project is the RAS MAPK signaling pathway. Noonan syndrome is caused by enhanced signal flow through this pathway. The signaling cascade mediates biological functions, including things like cell proliferation, survival, fate determination, and differentiation. It is activated in response to cytokine, hormone, and growth factor stimulation. So, activating mutations in 5 genes coding for transducers and modulator proteins participating in this pathway, so an example being SOS1, account for about 1/4 of Noonan syndrome cases, and missense mutations in node SOS1 account for a large portion of cases as well. Something interesting. So, involving targets for therapy, Noonan syndrome leads to very much increased activity and activation of the RAS MAPK pathway. The and the improved understanding of the molecular mechanisms that underlie the different manifestations of Noonan syndrome and the RASopathies, I think that's how you say it, has led to the identification of various molecular targets for specific pharmacological interventions. So specific agents like SHP2 and MEK inhibitors are things that have already been developed for the treatment of RAS MAPK driven disorders such as Noonan syndrome. So the idea behind this is the any kind of therapy that's going to be suggested or developed is going to have, for Noonan syndrome, is going to have the goal of reducing RAS MAPK hyperactivation. So that's kind of the idea behind these targets for therapy. And then in conclusion, in this specific scenario about regarding Noonan syndrome, Systems Bio serves as a very useful tool in studying the interactions between various genes, proteins, and pathways to either come to conclusions or just to explore more about how different components in the system affect each other, which was seen in the way the SOS1 and the pathway, the RAS MAPK pathway, kind of affected each other. Again, this project primarily focused on how SOS1 affected and was affected by the pathway, which is the main one involved in the development of Noonan syndrome. Here are my references, and thank you for listening.